We will wait for 5 more minutes and then we shall start the webinar.
Okay.
Hello and a very warm welcome everyone to our today's webinar on data safety assessment in clinical trials, an overview of CDISC, SDTM, AE domain and safety reports. I am Yuvragi Chandeli and I represent Veeda Clinical Research Limited. Veeda Clinical Research, together with its subsidiary BioNeeds India Private Limited and its joint venture Ingenuity Biosciences Private Limited, together referred to as the Veeda Group, offers a comprehensive portfolio of clinical, preclinical, and bioanalytical services to support innovator, biosimilar, and generic drug development programs of our global clientele. Together, we serve our clients globally in the following industries: pharmaceutical and biopharmaceutical, agrochemical and industrial chemicals, herbal nutraceuticals, and medical devices. As the facilitator today, I'd like to introduce you all to a couple of points as we begin this session. The session will have a presentation in the beginning, followed by a panel discussion at the end. For all your questions and queries, we have enabled our Ask a Question feature. It's right on your screen, so you can drop your questions in there. Moving on, I would like to introduce you all to our presenter for today's session, Mr. Koteshwar Yadam. He's presently working as Senior Statistical Programmer with Veeda Clinical Research. Mr. Koteshwara Rao completed his BTech in Biotechnology and PDGPM in Biotechnology and Management. He has over 7 years of vast experience in statistical programming. His expertise lies in CDISC implementation, which includes SDTM and ADM, and preparation of safety and efficacy reports for both BAB and clinical trials. He worked for various pharma and CRO companies prior to joining Veeda Clinical Research. Without any further delay, now I would like to virtually call upon Mr. Koteshwara to start with his presentation. Over to you, Mr. Koteshwar.
Thank you, Yurangi. Thank you so much. Hello, everyone. Good evening. Um, so today we would like to discuss about the clinical data safety assessment in clinical trials and an overview of CDISC, SDTM, AE domain, and safety reports. Okay. So I would like to discuss about CDISC definition. So CDISC is Clinical Data Interchange Standard Consortium. It is a organization which was formed in 1997 by group of volunteers. So CDISC is a global and open multidisciplinary non-profitable organization that has established standards to support the acquisition, exchange, submission, and archive of clinical research data and metadata. CDISC stands are vendor neutral, platform independent, and freely available via CDISC website, which is called cdisc.org. So CDISC is an organization which is having standards. The standards will be enabled to speed up the drug development process. The standards we are going to discuss about SDTM. In SDTM, we're going to discuss about AE domain. SDTM stands for Study Data Tabulation Model, and we have an implementation guide, which is version 3.3, and ADAM is stands for Analysis Data Model, and IG Implementation Guide version 1.2, and we have Case Report Tabulation, which is Define XML version 3.1. So, what is the benefits of CDISC when the sponsor is submitting to FDA? Okay, so having the standard, the sponsor can send the data uniform way, and FDA reviewers also can speed up the drug development process, and they can review the data efficiently and effectively. If we have a data standards, then which will be helpful for the sponsors and FDA reviewers. And we can also speed up the drug development process, and it can be also help the sponsors to bring the drug to the market in a shorter duration. So review time is less by FDA reviewers, and CDISC is open and multidisciplinary and non-profitable organization. So all the standards are freely available from the cdisc.web or cdisc.org. We can download all the guidelines from the website, and then so they can save the FDA reviewers also can save the time. So I would like to introduce here the CDISC implementation flow. So the flow we follow at Veeda. So first we will receive the study data after the database, and then we will actually check each and every domain according to the implementation guide version 3.2 or 3.3, and then we will prepare SDTM outputs. And then again, once we created SDTM outputs, we'll prepare ADAM outputs based on the ADAM implementation guide. And then we can also prepare the tables and listings and graphs based on the mock shell. So this is the implementation flow. We will be using SAS 9.4 software. So the guidelines we'll be using here depends on the sponsor. We can use for studies 3.2 or 3.3, and now the current version is 3.4. If the sponsor is required 3.3, then we can go for 3.3 guidelines. Okay, and then so first what we do is here we will create a specification, so which is nothing but a programming plan which will be implemented in the Excel, and specification is a functional document which will guide us how to do the programming. So then we will prepare the annotated CRF as for the guidelines, and then we will prepare a programming part. So we'll do validation and the development. Both we do two-way programming during the implementation of SDTM. So SDTM domains will be created based on the protocol specification and ACRF and the implementation guide. And then once we created the outputs, then we will prepare a define XML file by using the Pinnacle 21 Community 3.1.2 guidelines. So then we will prepare the reviewer's guide. So before we prepare the reviewer's guide, we do validation. We do the external validation from the Pinnacle 21. So based on the Pinnacle 21, we can resolve the issues. If we are not able to resolve the issues, then we will discuss in the reviewer's guide. So which will be implemented same for the ADAM analysis data models. Same process will be implemented for add-ons. This is the workflow we do follow at Veeda. And according to the implementation guide, 3.2 SDTM domains are classified into special purpose category domains, interventions, general observation class, and events and findings about and trial design models and some of the special purpose, which is RELREC and supplementary qualifiers. So what CDISC is created some standards. So the standards are based on the general observation class. Once the clinical trial is started, from beginning, from screening to enrollment to end of the clinical trial, we'll get the data. So based on the data, we will map the clinical trial, the study data into different domains. So the CDISC was classified the different domains. Now today we will discuss about AE domain, which comes under events category, events general observation class. The AE domain will capture the adverse events, one record per adverse event per subject. So AE domain includes clinical data describing any untoward medical occurrences in a patient or a clinical investigational subject administered a pharmaceutical product, and which does not necessarily have it to have a causal relationship with the treatment according to ICH E2A guidelines. The events included in the AE domain should be consistent with the protocol requirements. Adverse events terms may be captured either as free text or via pre-specified list of the terms. So according to the protocol backend from DM team, and they will create a CRF, and according to that, they will follow the CDISC guidelines as well. And according to that, they will collect the data. Okay, so why do we collect the adverse event data? So to determine the safety profile of the drug. So to evaluate the risk and benefit of the product, and to provide the information approval for marketing. Determining of safety is often one of the primary protocol objectives when evaluating the new therapies. And protecting subject safety is one of the most important responsibility of an investigator. So we do collect the data for every primary endpoints of the protocol. So safety. So safety, which includes adverse event, and lab and demographic vitals physical examination also. Okay, so these are all the factors why we are actually collecting the adverse event data. And then examples of the adverse events. So adverse events are side effects. Okay, and so it could be abnormal lab values and physical signs or symptoms, and abnormal exam test or procedure results, and concurrent illness or change in the vital signs or physical exams or fever. Okay, so during the clinical trial, the investigator will assess what are the adverse events are experienced from the subject, and they'll write it on the CRF page. So serious adverse events, which is serious adverse events are when the adverse event is worst, then we can call it as a serious adverse event. The serious adverse event might be leads to the death or life-threatening or hospitalization or congestional anomaly by birth defect. Okay. Um so as we know, the CRF case report form, which will be created or designed by the DM department. Um, so according to the guidelines, CDISC guidelines. So here I will discuss about CRF, how the data we will be receiving from the CRF as a source. Okay, AE number, adverse event term name, and then did the patient reported this AE after the dosing, yes or no, start date, end date, and ongoing, whether the adverse event is ongoing, and seriousness of the adverse event, severity, causal relationship, and then action taken, treatment for AE, outcome, and serious adverse. These are all the just general terms, so general considerations from the CRF, which will be checked from the every patient during the study phase. So once the database is locked, we will receive the raw data in the form of Excel or SAS XPT file. So we will analyze, we will check every column, every variable, every value, and then we'll map it to the SDTM guidelines, CDISC guidelines. Okay, so for example, if you see here, adverse event is start date, adverse event start date, AESTDAT, but our standard variable name is AESTDTC. So according to the standard, we will rename the variable, and then we'll also change the date format. According to this, on the data we are receiving from the database data management 10th May 2021. So we will format the dates according to IS 860 DA dot or DT dot. So it is year, month, and date. So I'll show you the how we will get the format of the dating. So we follow the specification, which is SDTM implementation guide. So we have a standard variable names. We will actually check each and every variable name, and we'll try to map to the raw data which we are getting from the DM department. Okay, so I would like to read some of, I would like to go through some of the variables. We have around 50, variables we have. So I will discuss very important variables. The AE term, one of the first variable, AE term, reported term for the adverse events, which is a topic variable, which is very important in the domain. So we have to populate, verbatim name of the adverse event, name of the adverse event. Here we will see the every variable name, standard STTM variable name, variable label, and then type, and then control terminology or core list or format, and then core. When you see here required, so definitely we have to include it in the domain. When the variable core is expected, the variable must be included. The values can be missing or not missing. When it is a permissible, so if we have a raw data, then we will include, otherwise we will drop the variable. Okay, so the AE term is the topic variable, and then backend, the DM department, they will also use the MEDRA. They will actually, use the MEDRA for the adverse events, the lowest level term, lowest level term code. So every adverse event, so we have a medical dictionary. So we will get the lowest level term and the highest level term, body system organ class. We have some columns we'll get. So we will map to the as per the AE domain variables. So apart from that, the severity of the adverse event. The severity is mild, moderate, and severe. So based on the investigator, the decision, so the findings, they will write down the mild or moderate or severe adverse event. Okay. A is serious. Whether if the adverse event is serious, then the serious adverse event is Y. Okay. And then action taken, any action taken by the investigator. Let's say subject is having the fever. So you may prescribe one medicine, let's say Dolo. So what are the action taken by the investigator during the study phase because of that specific adverse event? And then causality. Causality is related to the study drug or not related to the study drug, based on the investigator decision. So we'll get the data related or possibly related or not related or unlikely related or certain. So this type of terminology we will receive. And then there are some serious adverse event related variables, which are is that serious adverse event is leading to cancer or death or disability or hospitalization. This type of the data we will receive, and then we will check each and every variable, standard variable name and label and the values, every variable attributes. According to that, we will include it in the main domain. And then couple of variables which are related to the timings, when the adverse event is started, date and time, and when the adverse event is ended, that means resolved. So we'll see this type of variables in the timings, and we'll also follow the control terminology, STTM terminology for whichever the variable having the control terminology. and we must follow the control terminology for every variable values. Let's say outcome of the adverse event. One of the variable called A out. So outcome, so the values are fatal or not recovered or not resolved or recovered or resolved. This type of terminology we use when we are doing. So we will check each and every value, and is that value is compliance with the CDISC control terminology, and then according to that we will populate the values. And this is the raw data. We have raw data. So here raw data. So from the backend DM, let's say for example here CAU. If we see the values like unlikely or possibly, so we will rename the values to our standard variable name AREL. So this is the mapping process. We do programming, SAS programming, and then we will create values and variable names. We'll rename the variable names. Finally, we will get the output like this. So here we will actually create a sequence number, and then so we have a study ID, domain, use subject ID, sequence number, sponsor ID, term, a term, modified term, and lowest. These are all the Medra related variables. Okay. Once we have completed the AE domain, so all the SDTM domains, so we will validate from the Pinnacle also. So by preparing the define XML file, and then before that we'll prepare metadata. From the metadata we'll prepare define XML file. From the define XML file we will prepare the final validation. If we have any issues, then we'll discuss in the SDRG, Study Data Review Guide. So then for the safety analysis, we will also go for analysis data adverse events. So here what we do is we will construct the AE domain. Based on the AE domain, we will use some population flags, and then we will use some treatment related variables um, for the generation of ADAE domain. So we'll actually merge some of the treatment related variables and then some of the population flags, safety flag, okay, complete analysis flag, okay, and then per protocol flag or intended to treat flag, and some of the treatment related variables we will map to the AE domain. So then we will prepare the ADAE domain for analysis. So based on this ADAE domain, we will create the tables, so which are part of the safety and safety evaluation. So we have some tables. I have given the examples. Overall summary of treatment emergent adverse event. So the treatment emergent adverse events are nothing but subject after the study treatment at least one dose, any medical occurrences. Okay, so then we will call it as treatment emergent. Um, so the here we categorized into subject category and treatment name, test and reference and total. So we'll here we will prepare, we'll make a count number of events for the test, how many for test, how many for reference, and how many for total. And similarly, we will also calculate the count and percentage for every subject level. So subject, if any subject, number of subjects with at least one adverse event. Okay, if number of subjects, we'll make a count for test and reference, and the total for at least one severe adverse event. So based on the mock shell, so we will prepare the count and percentage. We'll calculate the count and percentage. We'll prepare the table, and then this is one of the other table, the incidence of adverse events, relationship with the treatment. So how many subjects are experienced adverse events because of the study drug. So we'll make a count for test and reference, and then total, and then system organ class. Each and every adverse event, so is cause, the system organ class will be nervous system or circulatory system, or any adverse event which is actually affecting this which system organ class. System organ class and then preferred terms. So first system organ class and then preferred terms, followed by the preferred terms. System organ class 2, preferred terms. So we'll calculate all the count and percentage for every system organ class and preferred term. And we'll also discuss footnotes related to the adverse events. If the sponsor, before we go to programming, so we will check with the sponsors also. So according to the sponsor's requirement, if we change this, then we can also change the the mock shells. So here the safety reports, which will be very crucial for regulatory perspective also. So IND safety reports. So a report issued by the sponsor of an investigational product when the safety issue arises. So they will submit all the adverse event, the safety related data, adverse events and lab and related test information to the safety. So we have to submit it to the FDA and investigators and IRBs also. And it is also mandatory that the regulatory regulations 21 CRF 312.32 C1. So safety analysis also very important in the clinical trials. So they will actually submit to the sponsor side. They will submit to the all the regulatories. And then apart from this, interim analysis during the study phase, DSMB, Data Safety Monitoring or Board Committee. So an independent committee of clinicians, statisticians, ethicists, and other specialists who access the progress of the trial, its safety and or its efficacy at specified intervals. So we do prepare some adverse event related tables according to the sponsor for the DSMB submission for interim analysis. So then what based on the reports, the committee can make recommendations that a study can be continued or modified or stopped based on the data reviewed. So this is what we actually prepare DSMB reports, some of the studies based on the requirement. So that's all from my side, and these are all the couple of references I have given. Uh, Over to panel channel, Dr. Kansham Patel.
Yeah, hi Kodeshwar. Yeah, nice. The presentation and everything is fine and everything is good. Yeah, now we collected some questions from the audience and all. Yeah, we'll take care of questions and all. Prasad here.
Hello,
Um,
Prasad.
yeah. Yes, what is mean by toxicity grade and any document will be used for this?
Yes. So toxicity, we will actually use the common criteria for adverse events. So we have current version is 5.5, 5.1. So then we have one document which will be used for the common terminology criteria for adverse events. Okay, so this is the 5.0. 5.0. So basically five types of grades we have, grade 1, grade 2, grade 3, grade 4, and grade 5. So grade 1 is the mild, grade 2 moderate, and then grade 3 severe, grade 4 life threatening, and grade 5 death. So based on the adverse event severity, based on the investigator decisions, so they may use the CTCAE, and then we will receive whether the grade 1, the adverse event is belongs to grade 1 or grade 2 or grade 3 or grade 4 or grade 5. This is the documents actually back and they will use.
Okay, okay. What is the code list value and give me one example of on that.
Okay, code list is the SDTM related terminology.
Mhm.
So for every variable, some of the variables we have a code list. Not all the variables, some of the variables we have a code list. Whenever we have a code list, we have to definitely check the value exactly. Okay, so let's say the male and female is the value. Okay, but according to the code list, we don't populate the male. Instead of male, we populate M value. Instead of female, we populate the F value. So we do have some control terminology for some of the variables. According to that, we will populate. Let's say this outcome, I have given the example outcome recovered or resolved. So apart from that, if we go through the SDTM terminology, so this is the SDTM terminology we use for the control terminology check. So here, let's say we take action taken. Okay. So this is what we have. The C66767 is the code, and then code list. These are all the code list names. So during the action taken variable, we have AEACM. Let's say here the values we have to populate exactly based on the CDISC definitions. This is what we call code list for one of the variable. So code list is nothing but so sponsor, whatever the values we are receiving from the sponsor, the value can be changed according to the these guidelines.
Yeah, okay. One more question. Any AE table related to serious adverse events?
Yes, we do populate a couple of the tables. We will actually mention serious adverse event also. So here you see number of serious adverse events, and then even subject level also. So subject level, number of subjects with at least one treatment related serious adverse event. Okay, so yes, we do actually calculate the count for the reference, and the based on the test, we will also calculate the serious adverse events. Yes.
Okay. Okay. What is the number of events on unique or totals? What is this?
Okay. The here what we do is the number of events we will be calculating based on the distinct of the adverse event term, preferred term. Okay, so here let's say the whole study we have 1,000 records, and the adverse events are let's say 200 only. So this is the 200 is the distinct. If the sponsor want to include the total number of adverse event, we will prepare the total number. If they want a number of events, then we can include the number of events, which will be the distinct of the total adverse events.
Okay. Okay. What do you mean by study guide?
Okay, study review guide. What we have is after the validation of the Pinnacle
Hmm.
21. So we will actually use the Pinnacle 21 guidelines community to validate the SDTM outputs and ADAM outputs. Whatever we can do, the programmatic or data issues, programmatic, so we'll resolve it. If it is related to the data, then we'll send it to the DM department, and then we will actually receive anything will be resolved. If not, then we will actually create a guide which is called SDRG, Study Data Review Guide. So study data review guide is a guide which will be discussed about every what are the issues come across during the validation.
Okay. Which version of implementation guide you use to generate the EE domains?
All the domains, all the STTM domains, we will use 3.3, STTM 3.3 version. If the sponsor want, then 3.2, we can also go for 3.2, or now currently 3.4. So now November 2021, they released series the STTM implementation guide. So if any sponsor is specifically suggested, then we'll go for any implementation guide.
Okay. Will you include all the variables from the EE domain?
All the variables it depends on the raw data we're collecting from the DM department. So we have a list of variables around 50 variables we have, but out of 50, whatever the raw data we have required and expected, we definitely keep, and if not, then we will actually ignore the variables. Okay, so it depends on the raw data. If we have a raw data completely, which can able to map all these variables, then we will actually include it. But required and expected variables, definitely we'll keep it. But coming to the core permissible, if we have any raw data or any variables values related to that permissible variables, then we will include. Otherwise, we will drop that variables. So it's not possible to completely include all the variables, but it depends on the raw data. If we have a data related to all the variables, then we can include. Otherwise, whatever the required and expected variables, we can include it in the A domain.
Okay. Yeah. How many adverse event tables will be created for the study?
It depends on the study. It depends on the sponsor also required. So as for Veeda standards, we have a couple of around six, seven adverse event tables. So it depends on the sponsors. If the sponsor want more, then we can actually make it. So according to our biostatistician team, so they will actually create the mock shells based on the protocol and in the SAP. So this type of tables, like you see here, instance of adverse events by treatment, irrespective of causality, and this type of tables, the serious adverse events related tables, and these are all the just couple of tables I'm just going to show you. And then every severity wise, moderate, mild, and severe, life threatening, every severity wise we will create. Okay, a couple of tables, like 6 to 7 tables we will prepare. Depends, and it depends again. So before we sign, so we'll discuss with the sponsor, and if sponsor required, we'll also include based on the biostatistician, they will also check the ICH guide
I'm
Q.
hand over to Jaideep for the further. Please take it.
Thank you, Dr. Gancham and Mr. Varaprasad, for your active participation in this panel discussion and nicely answering to most of our audience questions. I'd like to take a pause over here. We understand this session has kept us all well engaged throughout, but due to time constraints, we'd not be able to take any further questions at this point of time. But please be rest assured that we'll be sending the answers to each and every question of yours via emails. Also, we'll be sending across the recorded link of this webinar on all the registered email IDs. Lastly, I, on behalf of Veeda Clinical Research, would like to sincerely thank you all for being here today and taking out the time to be part of this webinar. I'd like to extend our gratitude to Mr. Koteshwara Yadam for being such a wonderful presenter and giving us all great learnings today. We at Veeda Clinical Research honestly look forward to host you all again. Till then, take care and stay safe, everyone.
Thank you. Thank you, everyone.
Yes, thank you. Thank you, everyone.